This week in lab, we're going to look at the microscope. Now, the microscope is an essential tool in the micro lab. I do know a lot of you have experience with the microscope from previous courses, so we're going to use this week to kind of review the parts of the microscope and their function. Please look in D2 well because there is a diagram or a picture of the microscope that you'll be using as part of your assignment, and there is a review list of terminology to help you as well. When it comes to the microscope that we use, you need to make sure you understand that it's compound, light, and bifocal. So a light microscope or bright field microscope just simply means that we use light sources to pass through the specimen, and it helps us looking at stained and unstained items and cells and various pieces, and allows us that visibility. It is considered to be a compound microscope because it uses two magnification systems. So it's got both the objective and it's got the ocular, so we compound those, and we'll look at that here in just a minute. And then it's considered bifocal because we have two ocular pieces. But again, understand compound, light, bifocal. Also understand all the terminology here and know those parts by looking at either this slide or go to the document that is downloaded in D2L, and also look at the review terms in the D2L. There's a sheet there for you. Make sure you understand magnification of the microscope and how it works in that compound magnification system, and the fact that we have to take the objective lens times the ocular lens to get the total magnification. Another terminology, make sure you understand that why we use the microscope. It's to help us with resolution, okay? And that means that we're able to take that minimum distance and be able to still distinguish between 2 points. Why this is important is because we're not just making something larger. Think about with your cell phones. If you've taken a picture on your cell phone and then you use your fingers to try to enlarge the picture, and sometimes you lose clarity and it becomes very, very fuzzy. In microbiology, we can't just afford to make it larger. We have to also make it larger with the ability to see between the dots, if you will, so that it has that high level of resolution. That's important in order for us to be able to use this tool appropriately. So again, we've referred to the fact that it is an dual ocular system, okay, and the fact that it's got objectives and ocular lenses, and the total magnification. So always know how you come up with a total magnification for a quiz question or on the practical, etc. And again, I believe most of you have had that many times, but just wanted to reiterate it. The other thing is that we oftentimes use oil immersion in microbiology, and the reason for that is it gets rid of the light noise, if you will. So oil immersion, if you use a drop of oil and you put it on the slide and then you use the 100x objective, it will then help concentrate all the light up through the specimen, and thus it increases the magnification, and even greater, it increases the resolution, so that we not only see it larger, but we see it with more clarity. So oil immersion is another part of this tool that we use more in microbiology than you may have used in other of your science courses. So just to kind of reiterate, understand that it's binocular or bifocal, okay? Parfocal, what it means is the fact that you put your slide on the microscope and you bring it into focus using the scanning objective of 4x, and then at that point you turn it to 10, and it should just take little bits of focus, very little bit, because of parfocal system, so very little moving to be able to bring it back into focus, okay? Resolution and magnification. So microscopes are only good, we need them to magnify, but only if we can do it with resolution to provide us clarity. Now, not being in lab, you still need to basically understand how you go through that process of focusing the microscope. So here's a good video. The link is also found in D2L in their week 2's module, but review it. That'll help you get a better understanding of the microscope, in particular on how we go about focusing it. So that's the third and final of the videos for week 2. Should you need any help on any of the materials, again, don't hesitate to contact me.